And before I hand over to our speaker today, let me just set the scene for liquid biopsy and how it can be used as a research tool. What is liquid biopsy? I'm sure you know. It's analysis of bodily fluids for tumor-derived material. And actually all bodily fluids can be analyzed for liquid biopsy, like urine, saliva, etc. But mostly and more widely, we speak about blood-based liquid biopsy. And when we talk about blood-based liquid biopsy, there are quite some analytes which can be found in blood, like circulating tumor cells, cell-free RNA, circulating tumor DNA, exosomes, and even tumor-educated platelets. Now, when it comes to application of liquid biopsy analysis, there are quite some which are in the clinic already now in the routine usage, like analysis of ctDNA for mutations, for patient stratification, for targeted therapy. Like, for example, very well-known example is T790M mutation analysis for a tyrosine kinase inhibitor prescription of the third generation. But this is not the limitation of liquid biopsy, right? There is so much beyond this clinical application, which is now in translational space and also in research space. And this is, on this topic, we would like to focus today. And actually why? Just because liquid biopsy is such a unique source of material from tumor. And given there are these multiple analytes in blood, you can get information on different omic layers, right? So genome, transcriptome, epigenome, proteome, and also maybe some other information. And why does it matter? Because we know that cancer cannot be fully explained by single omic. Cancers are regulated by the complex interplay between the omes and the microenvironment. And if you think even further, tumor heterogeneity plays a significant role in cancer development and progression, right? And liquid biopsy is a unique source of information when it comes to study of causes and consequences of tumor heterogeneity. Because if you think of conventional tissue biopsy, the information you can derive from tissue is limited to the time point when biopsy was taken and to the spatial context from where the biopsy was taken. And so you can miss information which is relevant for your study just because a certain clone might have been missed by the conventional needle biopsy. Liquid biopsy, in contrast, gives you a comprehensive view because all clones of the tumor will be represented in blood, and not only of the primary tumor, but also of metastasis, right? If you think about patients who have, for example, brain mets, which you cannot biopsy, this is where liquid biopsy is a very, very valuable source of information. So it enables comprehensive and real-time snapshot of the disease. And given it's minimally invasive for the patients, it's like just simple blood draw. You can do it sequentially and so often as you need. And this is why liquid biopsy became a very valuable tool in translational space in what we call forward translation, so from the bench side to the bed, right, when it comes to biomarker discovery, drug target discovery, and understanding of cancer biology, but also other way around, from bedside to the bench. You can do a lot with liquid biopsy to optimize therapy regimens, to maybe find other additional biomarkers and drug targets. And in this context, there is a very particular source of information in blood which can be analyzed, and these are circulating tumor cells. The cells detach from primary tumor or also from metastasis or from tissue of the tumor, from tumor material, and they can be found in the circulatory system. The lifetime of circulating tumor cells is very short, but the information you can derive from these cells is really valuable because the cells are viable, so you can analyze them on genomic level, transcriptomic level, epigenomic and proteomic level, but also you can do functional studies, you can do CTC-derived xenografts, and you can do drug testing. And with that, I will pause here and I will hand over to our speaker today, Professor Nikolas Stoecklein from the University Hospital Düsseldorf. Nick studied medicine in Hamburg in Germany and spent some time as medical intern in the UK. He did his postdoc at the Institute of Immunology in Munich and then moved to Düsseldorf, where he started his career at the Heinrich Heine University in Düsseldorf, first as resident in surgery, going the whole way up to professor for experimental surgical oncology. Nick is a member of multiple societies, including EACR and also European Liquid Biopsy Society. He's the author of more than 100 publications in PubMed, and his main field of research are circulating tumor cells, liquid biopsy methods, single-cell technologies, tumor cell dissemination, and minimal residual disease in solid malignancies. And with that, Nick, the stage is yours.
Yeah, thank you, Anna, for this kind introduction, and you gave also already a lot of information as a great introduction to my talk. This is so today I want to highlight the potential of circulating tumor cells, CTCs, and I hope I will give you some kind of new perspectives on this topic, especially because I'm convinced that we will need high blood volume sampling for bringing this eventually in the clinic. So this is my disclaimer. To start, give you a brief background on circulating tumor cells. So these are cancer cells that have entered the circulation either by active or passive mechanisms and can be traced. Oops, sorry, I have to manage here with my presentation modes. Okay, as very rare cells in the blood. It's, I think, important to notice that the concentration in the peripheral blood of CTCs is several logs below that of normal blood cells. Therefore, we need very high sensitive methods to detect these ultra-rare cells. One of the gold standards that we use right now is the cell search system. That system has been used in hundreds of studies and thousands of patients have been investigated with this. And what we have learned is if we find an CTC, which is actually an EpCAM-enriched cell positive for cytokeratins, then the patients have a poor prognosis. And the more CTCs patient has, the poorer the prognosis will be. However, beyond prognosis, there are several other potential clinical applications with potential clinical utility across the whole course of the disease of the patient. When you think of the primary tumor situation before surgery, sampling for CTCs can give information whether a patient has a higher or lower risk to develop relapse to tailor adjuvant therapies. And then it will be, or it could be possible to test the effectiveness of that. And it has been shown that the reoccurrence of CTCs after resection can precede the imaging by radiological standard methods several months. So this can be kind of an early detection system. Once the metastases have been established, the idea is to use these cells to isolate them and get information on the characteristics of the cancer cells to decide on the treatment and also to monitor then the effects, because this has also been nicely shown in prostate cancer that already 4 weeks after a treatment was initiated, you can already see whether a patient responds to the therapy or not. This is also several weeks before you can see something on imaging. And if the tumor is resistant, then of course the potential here is to isolate the cells, get an idea on the resistance mechanisms, and try to devise a new therapy based on the characterization of these cells. Surely these are all methods that can also maybe be addressed by circulating tumor DNA. The question is, what can CTCs add to that? And I think it's important to notice that it has been formally shown that CTCs comprise the drivers of metastatic progression. That has been, for example, shown by Andreas Trump group from Heidelberg or Caroline Dives group in Manchester. And most importantly, the CTCs contain the complete inventory of all clinical relevant analytes, proteins, DNA, mRNA, and so forth. And beyond that, you can also get morphologic information, for example, on the expression pattern of a target. So from my perspective, CTCs are the perfect tissue surrogate to perform predictive tests and do monitoring studies. And to give you a glimpse of that, I want to give you a first example for such a study that was conducted by our close collaborating group in Düsseldorf, Tanja Fame's group from the gynecology department, Hans Neubauer, and most of the work was done by Andre Franken. And the idea was here what kind of clinical information you can get if you do multiparametrics analysis of those CTCs at different time points during the treatment. And they focus, of course, on metastatic breast cancer patients with high CTC counts, which means over 90 CTCs in 7.5 milliliter of blood. And one of the first things they noticed after isolating those CTCs and doing whole exome sequencing was that they found in actually all of those patients they investigated mutations relevant for targeted therapies, and importantly, often these mutations were not found in the matched tumor tissue. They followed one patient, patient 1, very closely over the whole time of the treatment, isolated CTC samples, and actually at time point 3 months, they were able to generate also a cell line derived from these circulating tumor cells. Their whole exome sequencing study showed that there were 8 clones present, mostly in all of the metastatic tissues and in the primary tumor. Of course, that was, I think, quite interesting that they could follow here also the dynamics. And in one of the clones, they found particularly of interest, clone 8, which they devised as the kind of resistant clone that seemed to drive then the disease in the end. They found an ACT1 mutation, which which the CTCs carried at all the time points. And this, together with the phosphorylation of ACT, could be a potential therapeutic target for ACT-directed therapies or mTOR-directed therapies. So because they have had so many cells at these different time points, particularly also they had this CTC cell line, they could check the phosphorylation status of ACT on CTCs and found that the majority of those cells actually had activated ACT. And then they could also do sensitivity studies on the CTC cell line derived from that patient and could see that, for example, everolimus would be a suitable therapeutic option. And after futile treatment with paclitaxel here in the at time point 33, the doctors decided to give it a shot with everolimus based on those data they obtained from this patient. And in fact, the CTC numbers went down significantly, and the patient had then stable disease. So this is, I think, a quite interesting example, well, exemplary, of course, that could show what the potential is, how you can follow the patient's disease with CTCs and then devise the therapy. So in this context, I will present you another study that is not published yet. here the idea was to use single-cell CTC information to investigate resistant mechanisms in prostate cancer. And the background here is the PARP inhibition therapy with olaparib. This is actually work driven by Johan de Bono's group at the EACR in London. They brought that really into the clinic, and they found that this has anti-tumor activity and that the patients benefit from olaparib therapy, metastatic castration-resistant prostate cancer patients. However, they need to be selected based on these DNA damage repair genes. So you need to get prior information before you give this therapy. However, despite the selection based on these genes, around 50% of the patients do not respond to the therapy. So the idea was to use information from CTCs to get an idea what could be the resistant mechanism here, what is the heterogeneity of the CTC population under therapy, and can we assess the clonal dynamics under therapy. This is the workflow we used, and we have published some years ago. So after cell search enrichment, we perform isolation of single cells, either by FACS sorting or with a DEP array. If we have the single cells, we amplify the whole genome using adapter linker PCR, the APLI1 method, and then we can use those amplification products to obtain copy number alteration profiles. In the beginning, we did this with array-based comparative genomic hybridization. Now we use low-pass next-generation sequencing for this. So in the clinical study, blood samples were taken at 3 time points in the UK: baseline, under treatment, and at the end of the treatment. The CTCs were determined in the UK, and then the cartridges of positive patients were sent to us in the lab, and we then isolated the cells. We could isolate more than 1,600 cells, and around 70% of them had high quality to perform such comprehensive genomic analysis on the single-cell level. When we have a look at the CTC numbers that we got from the patient that we could investigate, we focus, of course, on those that had around the baseline 40 or more CTCs in the blood to have enough cells and enough power to do these analyses. However, in the patients that then turned out to respond to the therapy, you can see that they all showed a decline of CTCs with less than 5 CTCs at the end of treatment, whereas the non-responders remained very high CTC numbers. When we then did the genomics, first with array-CGH, we could observe that the percentage of aberrant genome of the patients, of the CTCs of the patients that responded, was significantly higher than in the non-responders, which is intuitive, I think, in this PARP inhibition therapy. The hierarchical cluster analysis also separated the CTCs from responders compared to the non-responders. So that was, for us, interesting to see, but of course we were much more interested in what is going on during the therapy, especially in the non-responders. And for that, we sequenced a relatively high number of cells per patient and obtained high-quality traces in those single CTCs. Those are the ones you can see here that we selected based on the quality that showed high quality. From those, George Seed, bioinformatician at ICR London, was then able to perform a phylogenetic analysis and to determine the number of clones that we have present in the blood of the prostate cancer patients. And it was interesting to see that these clones were present at all time points, so it didn't change. We didn't see anything new coming up or really that the therapy erased a certain clone. However, what we could observe, if we then look at the dynamics over time, which you can see here in these fish plots, was that we had two different patterns. One was that there was a predominant clone that persisted throughout the course of the treatment, whereas in another group of patients we found a minor subclone that could increase significantly under the treatment. In the next step, we tried to make sense of these genomic analysis, which is not so easy with these copy number information. However, we tried to do some pathway analysis here, and preliminary data show that here the FGFR pathway could play a role, but this needs to be, of course, further investigated. So these are two examples. What kind of information you can get from these CTCs. And I think also taking into account the whole literature that is existing, we have robust protocols to investigate these few cells that we can identify in the blood. We can assess the heterogeneity. And this was, I think, the work by Andre Franken did show that also a patient selection could be possible. However, the big problem is with the CTCs that you need to have sufficient CTC numbers. And of course, all these studies I've shown you were done on patients with large number of cells, more than 40 in our study. In the study from our gynecology partners, there are even 90 CTCs or more. But this is actually a quite rare finding. We have to consider that the majority of patients, even the M1 situation, has less than 5 cells or are even negative. And this is, of course, a problem if you want to rely on that and make clinical decisions. You want to have CTCs in almost every patient. And there are, of course, several explanations for this. One that you often hear is that you don't have the right markers to identify the cells or they're not the right technology. But I think the most important factor is that the volume that we sample is just not representative for the 5 liters of blood that is circulating in the patient's body. So imagine you have 1,000 CTC, EPCOM-positive CTCs circulating in the blood, and you want to use cell search to find those EPCOM-positive CTCs. So you take 7.5 milliliter of blood. If you do the math, you should have 1.5 CTCs in your sample. However, the CTCs are not normally distributed. They follow rather a Poisson distribution. And also, our assay is not 100% effective. And all that contributes to the fact that we have just a chance of around 30–35% to find one of these 1,000 CTCs circulating in blood. To increase the chance to 95% to find just one CTC, you have to take around 6 blood samples. At least this is what comes out from modeling. So you have to increase the volume of your investigated blood significantly. And the group of Leon Terstappen, Frank Kumann's work, he asked the question, what would happen if you could screen liters of blood? And if you could do this, regression analysis shows that you could find 10 or more CTC in almost every metastatic cancer patient. So this would be, of course, the way to go. But how do you do this without harming the patient? Well, we think that leukapheresis is here the answer and can help. Leukapheresis is a standard routine technology to separate and collect white blood cells from liters of blood. This was established already in the 60s to remove leukemic cells from the patient body, and it has been quickly adopted in the era of stem cell transplantation, and this is now used for that routinely. Basic principle is that the patient is connected to 2 lines, an inward and outward line, to this leukapheresis machine. Basically, this is a centrifuge. The blood runs here through this tubing system, in which, due to continuous centrifugation, counterclockwise centrifugation, the plasma is separated from the Buffy coat and this from the erythrocytes. And now the operator can select for example, mononuclear cells relatively precisely from the system. And the other aspect, which is very important, is that this is a harmless method done millions of times worldwide without serious side effects. So what is happening after such a leukapheresis? So you generate a blood product which is enriched for certain white blood cells, mononuclear cells in particular. After 1 hour, which process around 2.5 liter blood, you have around 150 times more of those mononuclear cells in your blood product. And why should that help? Should that help us with circulating tumor cells? Well, we don't know the exact density of circulating tumor cells, but we have a lot of hints, and they all point to the fact that these epithelial cells share a lot of biophysical characteristics, specifically density, with mononuclear cells. So the idea would be that if you perform leukapheresis, that you then can co-collect epithelial CTCs together with your mononuclear cells. And this is something we have investigated since many years now in Düsseldorf, and we called that diagnostic leukapheresis to separate it from the therapeutic leukapheresis done for stem cell transplantation. And very important here is Johannes Fischer, with whom I developed this approach, and of course our fantastic teams from gynecology and from my surgical department. So in our first study a few years ago that we did, we compared a peripheral blood draw with the leukapheresis that was done after the blood draw. So we ran the leukapheresis for 1 hour and processed around 2.5 liter. Got this product with 2.5 billion, containing 2.5 billion white blood cells. This is, of course, very difficult to process, so we restricted this to around 5% to feed this in the standardized cell search system, and then compared the peripheral blood with the DLA product. And what we found was that although we just analyzed a tiny bit of the leukapheresis product, we already saw a significant increase in CTC numbers, but also, and that was important, of course, in the detection rate. This has then been validated by cooperation partners independently or working together with them in breast cancer, prostate cancer, and other cancer types, lung cancer. And what we can say now is that there should be sufficient CTCs really in every metastatic cancer patient. It is a clinical safe method and should be integratable into the clinical routine. At least these teams that did it so far were successful. Question is, of course, whether then DLA can really improve the profiling of CTCs. So genomic analysis, but also maybe functional analysis, because we have, in theory, much more of those cells. So the first investigations focused really on the functional characterization, and in fact, this is also work by Andre Franken in breast cancer. Here he could show that from DLA products, it was much more likely that the CTCs grow in culture compared to the peripheral blood samples. And because he had so many cells, he could also at least get a glimpse in some of the mechanistics, and this is apoptosis. So we found that many of the CTCs were apoptotic, and if there were less apoptotic cells, they more likely grew in culture. Second interesting study done a few years ago is also from Johan de Bono's group in prostate cancer. Here, that led to, DLA led to a strong increase of CTCs, which were then used for genomic profiling. At that time, we did ARRIS-CGH, so we got a deep insight into the genomic landscape of prostate cancer that is present in blood, and we could find and investigate the heterogeneity that was often missed in the bulk biopsy of matched samples. And importantly, they also could establish a functional model, an organoid, from a prostate cancer CTC. And this is some pattern we have here in these studies. This is another, I think, very interesting work from Françoise Farras' group from Institut Gustave Roussy in Paris. Was published in Nature Communications. They were even able to generate a CTC-derived explant a model from a diagnostic leukapheresis of a prostate cancer patient, an advanced prostate cancer patient. And the comprehensive genomic analysis of CTCs, they did exome sequencing on single CTCs, on bulk tumor, and also on the cell line, helped them to understand the evolution of this particular case. And also, I think this was quite an interesting aspect. The model they established recapitulated quite nicely the sensitivity and the resistance to drugs they saw in the patient. For example, it responded quite well to taxane, but not to an androgen receptor inhibitor, to enzalutamide. So this idea of functional models generated from CTCs with the help of leukapheresis was motivating the team from the Erasmus Medical Center in Rotterdam to investigate this more in detail. And this is done by the group of Martin Lockema and Lisanne Maut. She was a PhD working on this. So they really asked the question whether they could regularly get these models out of their DLA samples. So for this, they screened over 100 advanced prostate cancer patients and wanted to include those that have more than 5 CTCs and 7.5 milliliter blood in the cell search analysis. And finally, they could perform DLA in 41 patients, and they tweaked a little bit on the leukapheresis protocol, so they actually increased the density of the cells they collected, and they observed in prostate cancer that seemed to have a positive effect on the collection efficacy. So they could increase quite heavily, actually, the CTCs in their DLA products. In the end, they calculated that more than 5,000 CTCs were present in their DLA products. They then depleted 25 milliliter of these products and were able to generate CTC-derived organoids in 35% of their samples. However, most of them didn't grow further than a few weeks, but in one case, they were also able to establish a permanent cell line, which then again recapitulated quite nicely the characteristics of the patient in terms of a response to therapy. So I think these are kind of, for me, impressive works that you can imagine that you have these circulating tumor cells, you get them out of the patient's body, and then generate these very informative models out of that. However, how could that be used in the clinic? Because from these data, it's clear that this is nothing you can do on a routine base. However, these data, especially from the Rotterdam group, clearly show that you can get in almost every patient a high number of vital cells that you can use for diagnostic tests. And if you consider that in prostate cancer you have now increasing options for target therapies in the advanced stages, and there is a clear need to gain molecular data to select these therapies, and you consider that bone biopsy is right now the standard for that, then DLA is a much more comfortable way to do this, really, and delivers high numbers of vital cancer cells, whereas in bone biopsy, which is not particularly dangerous but very uncomfortable for the patients, it fails in 25% because the material is just not good enough for molecular profiling, like DNA profiling, for example. However, we have, of course, big challenges still, and A major challenge is that we created a large haystack, which we have to process, so these billion of white blood cells. So right now we analyze as a standard 5%, and we can deplete more for these functional analysis, but we want to efficiently analyze, of course, 100% of the whole DLA product. And so far, we also don't know whether the extrapolations we do on our measured CTC counts are really true. So that was a question we addressed in the Cancer ID Consortium, where we analyzed at least up to 30% of the DLA product in lung cancer. And here we actually had a teamwork where 6 analytical sites independently analyzed or did a cell search assay on such a DLA product. And what we could see here was that we indeed have a significant increase of the detection frequency, but also of the CTC numbers, the more we increase the analyzed blood volume. But here, max, we are at 30%. We need to get to 100%. And this is right now still difficult. We have tried some technologies, depletion, different biophysical methods, or filtration devices. Problem here was that they're all not really effective, or it is difficult to work with these cells afterwards. However, there's a nice perspective that came out 2 years ago. The group of Mehmet Toner and Dan Haber at Harvard, they published their new chip, called an LA chip for leukapheresis, because I think they also believe now that you really have to sample larger blood volumes. And their chip-based technology, they claim that within 3 hours they can efficiently process a whole leukapheresis blood product. However, so far this was just done with model cell lines, so we have to wait until this becomes available, but I'm surely looking forward for this. So finally, there's of course the question, leukapheresis cannot be the only technology. Are there alternative strategies to enable high blood volume screening for CTCs? Well, there is the Gilupi cell collector that is claiming this. This is a device, a catheter that is brought into the cubital vein, stays there for 30 minutes. This catheter is coated with anti-EpCAM antibodies, and the idea is that the blood runs continuously over this catheter and collects the CTCs that are flowing by. We have investigated this in a single center study and compared this to cell search in colorectal cancer, and here it did not outperform really cell search in terms of numbers, and modeling, physical modeling indicated us that the investigated blood volume by this approach is around 10-50 milliliters, but not in the liter scale as that was suggested by the people that invented this. There's an upgrade to such a catheter. There's one group that proposed to have a magnetic wire, bring this into the cubital vein, and combine this with antibodies attached to magnetic particles that are also administered to the patient, and then if in vivo there's a reaction, these can be collected to the magnetic catheter. However, this has been just done in a porcine model so far, and I think it's quite far away from clinical practice. A quite interesting approach is this wearable device here from the Michigan group. And here the idea is that you wear such a CTC detection machine all the time for like 24 hours, and blood continuously runs through a chip, EpCAM-based, for example, and then collects those CTCs until it's full, and then you just take that off and can analyze that. This is right now not very effective, but surely this is, for me, a very interesting perspective for the future. and shows me that the people have great idea to improve CTC-based liquid biopsies. So to come to a conclusion, I think that high volume blood sampling is really the way to go because it can overcome the low frequency problem in CTCs. Of course, in patients with enough CTCs, you don't need to do this, but this can close this diagnostic gap that we have right now. And for the time being, diagnostic leukapheresis seems to me a feasible method. It's clinical safe, that has been shown, and the co-enrichment together with mononuclear cells that are targeted by this approach delivers enough CTCs for diagnostic tests. So we feel that this is a real liquid biopsy that can be done and should enable diagnostic tests based on CTCs in almost every metastatic cancer patient. Of course, technical solutions need to be refined, but they seem to be underway. So I hope the next 1 or 2 years or so, we will have a technical solution for that. And I would like to conclude my talk because I'm getting sometimes frustrated if you see all the new developments in circulating biomarkers and you still stick to your CTCs and it's going sometimes quite slowly. One of the ideas behind this liquid biopsy is, of course, to complement the fine needle aspirations that are the standard right now. However, if you read about the history of fine needle aspiration, it's interesting that when it was developed in New York in the 1930s, nobody cared, and most of the clinicians found it really a crazy idea. And it took 20 years that this was rediscovered by physicians at Karolinska, and then another 20 years that it became accepted and part of the managed care. So can take up to 40 years to bring something new into the clinic, something revolutionary like fine needle aspiration. So my hope is that we will also will be successful in bringing something good for the patients with a CTC-based liquid biopsy. And with this, I would like to thank you for your attention. I'd like to thank my team and our cooperation partners, especially members of ALPS. And I really want to highlight this European Liquid Biopsy Society. Please go on their website to get information for this. I think this is a very good society to stay informed. And I think here a big issue is also and a big topic to bring liquid biopsies into the clinic. With this, I'd like to thank you, and maybe there's time for some couple of questions from you. Thank you.
Thank you very much, Nick. Wonderful talk. We have time for a couple of questions. Other questions? Otherwise, let me start maybe with one. You spoke about molecular profiling of CTCs, and you touched on DNA analysis and also RNA analysis, so proteomic analysis. Obviously, you do protein analysis, right, because you stain your cells. So what's known about methylation analysis or epigenetic analysis of CTCs?
Yeah, I think this is, of course, something which has, I think, big future. We have one expert here sitting. I think for that, I'm actually thinking about this because now we have the possibility with DLA to get more cells for these comprehensive epigenetic analysis. We ourselves have done some single-cell experiments, which are quite limited, I have to say. So if you want to do more comprehensive profiling, maybe this will also help us here, diagnostic leukapheresis. And I hope that we can do this at some time point. But so far, we haven't done it.
Thank you. Other questions? Yep, please.
Hi, thank you for your presentation. The study of CTC have mainly focused on the study of single CTC, but now we know that the CTC can circulate with several cells in cluster, with other cancer cells or blood cells, platelets. do you know if the tools have been developed enough to study these CTC clusters in patients?
Well, I think the first study I showed you from Andre Franken that was published already there, they actually did a pooled analysis. So all the CTC they found and they isolated, they pooled together and then investigated this. What we did in the prostate cancer study, for example, we just focused on single cells. So the question of whether clusters are different, for example, we did not address. Actually, our sorting strategy is excluding clusters. So we just focus on real single CTCs to have the one CTC in our tube, which has also to do with the technology that we're using. With a dep array, that would be more easily possible, but we haven't focused on that specifically. But of course, on single cells, that is possible. So all the genomics that we're showing also apply to the single cell. If you have an amplification product, right now really you can do low-pass sequencing relatively easily, but also whole exome sequencing. You have to take in consideration, of course, that you have an amplification bias, obviously, and that this is just a representation of the single cell. So, and it's not like genomic DA, of course, but you can adapt your technologies, your downstream technologies to these whole genome amplification products. I think the technology is quite well developed to this point to allow these methods.
Thank you. We are running out of time, so if there are no burning questions, let's close the symposium. Nick will be around for a couple of hours. Feel free to ask questions, if I may say it on your behalf.
Sure.
And with that, thank you very much.
Thank you.